I want you to take a second and just like think about the physical space inside your head right now.
Right, just sitting right there behind your eyes.
Yeah, exactly. Encased in bone, you've got this mass of tissue weighing, I think it's about 1.3 to 1.4 kilograms. And I mean, it doesn't look like much, right? It's mostly just water, fat, blood vessels, and a whole lot of cells.
It really doesn't look impressive on the outside.
No, not at all. But that completely unremarkable looking mass is literally generating every single thing you call a thought, a memory, an emotion. It is generating your consciousness as you listen to this deep dive right now.
Okay, let's unpack this, because the very first people to actually write down observations about this mysterious mass inside our skulls were ancient Egyptian surgeons.
Yeah, we can trace that all the way back to the Edwin Smith Papyrus, which is from roughly 1600 BC.
Wow, that's old. Extremely old. And the author of that surgical text was, documenting head injuries and made a very specific, kind of strange observation. If a patient took a severe blow to one side of their skull, they became paralyzed on the opposite side of their body.
So hitting the left side of the head paralyzes the right arm.
Exactly. It's the earliest known record of what we now call contralateral control, which is just that the right hemisphere of the brain controls the left side of the body and vice versa. It was this massive clue to how our biology is wired, just sitting there in the historical record.
And the crazy part is humanity basically just ignored that clue for millennia.
Pretty much, yeah.
Instead of mapping out how the brain actually worked, the classical world got completely bogged down in this philosophical argument over whether the brain was even the thing doing the thinking.
Right, because around 400 BCE, you had Hippocrates arguing that the brain was the source of intelligence and emotion.
But then Aristotle comes along and says, no, the heart is the actual seat of the soul. So Aristotle basically thought the brain was just a radiator to cool down the blood from the heart.
A biological cooling system? Yeah, that was his formal argument. I mean, he looked at the brain, saw it was relatively bloodless compared to the heart, and just assumed it was a thermodynamic regulator.
What's fascinating here is how the debate was finally settled. And it required looking at actual biological mechanisms.
Not just philosophical debating.
Exactly. So in the 2nd century CE, a Roman physician named Galen, who treated gladiators, by the way, performed these extensive animal dissections.
So he's seen the anatomy up close.
Very up close. And Galen systematically proved that if you severed an animal's spinal cord, the connection between the brain and the limbs was broken, which resulted in immediate paralysis. But if you damaged the heart, the animal could still theoretically feel and process pain until the lack of blood flow finally caught up to the brain.
Which conclusively proves the brain is the control center.
Yeah.
But didn't Galen still get the internal mechanics, entirely wrong?
Oh, completely. He thought all the heavy lifting was being done by fluids sloshing around inside the brain hollow cavities, the ventricles. And medicine just coasted on that incorrect ventricular theory for over 1,000 years. It wasn't really until the 19th century that we finally started looking at the physical tissue of the brain itself as a map. And ironically, the catalyst for that was phrenology.
Yeah, phrenology. The pseudoscience of reading personality traits by feeling the bumps on a person's skull.
Which is totally bogus.
It was entirely unscientific, yes.
Yeah.
But, and this is key, it planted a crucial testable hypothesis in the medical community: localization.
Which is the idea that specific functions live in specific parts of the brain.
Exactly. That different mental functions are housed in different specific geographical neighborhoods up there. And that theory was finally proven in 1861 by a French physician named Paul Broca.
Right, Broca's area.
Exactly. Broca had this patient who suffered a very specific deficit. He could only speak a single syllable, the word tan.
That's all he could say, just tan.
Just tan. And when that patient passed away, Broca autopsied the brain and found a distinct physical lesion in the left frontal lobe.
A localized black spot that perfectly correlated with the loss of speech production. That's incredible. And then a bit later, in 1874, Carl Wernicke found a patient with basically the inverse problem.
Right, Wernicke's patient.
Yeah, the patient could produce language flawlessly, but had completely lost the ability to comprehend it. It sounded like word salad. And Wernicke found a lesion in a totally different area, the temporal lobe.
So Broca and Wernicke fundamentally changed how we view ourselves. They proved the brain isn't just this uniform blob of tissue where every part does the exact same thing. It's a mosaic of specialized modules.
A map of the self.
Exactly. And to understand how we function, we have to look at how that physical map is laid out, starting from the most evolved outer layers and then peeling it back to the primitive core.
Okay, so let's look at the brain the way a surgeon would, starting with that outer shell. The cerebrum is like the biggest piece of real estate in there.
By far.
And it's wrapped in the cerebral cortex, which is that deeply folded, wrinkly gray matter everyone pictures when they think of a brain.
Right. Classic image.
Yeah. And it's only a few millimeters thick, but because it's so folded up, if you somehow extracted it and flattened it out, it would literally be the size of a large pizza.
Which is a great visual.
Right.
What?
And we divide that pizza into 4 distinct lobes.
So you have the frontal lobe at the front, which handles your complex planning, your decision-making, and your core personality.
Then the parietal lobe right behind it.
Yeah, the parietal lobe integrates touch and spatial awareness. So basically how you know where your limbs are in space without looking at them.
And the temporal lobes.
Those are on the sides. They process hearing and memory. And finally, tucked away at the very back of your skull is the occipital lobe, dedicated almost entirely to processing vision.
So all of that wrinkly outer layer is gray matter, the actual bodies of the cells. And beneath that lies the white matter, which is basically the brain's cabling system.
Right, the wiring.
Yeah, insulated nerve fibers called axons that transmit signals between these different lobes. But before we dig deeper into those cables, we need to bust a massive textbook-altering myth about the cells themselves.
Oh, the neuron cat. Yeah, for decades, biology textbooks confidently stated that the human brain contains roughly 100 billion neurons.
Like, it was just accepted fact.
Totally. It was repeated in lectures, academic papers, popular science everywhere.
Until 2009, when a neuroscientist named Susanna Herculano Husel realized nobody had actually counted them properly.
Because counting 100 billion of anything is notoriously difficult.
Right. So she developed this wild new method. She took donated brain tissue and essentially dissolved the cell membranes, turning the tissue into this uniform chemical soup.
Brain soup.
Literally brain soup. And from that, she could isolate and count just the cell nuclei. And it turns out we do not have 100 billion neurons. We only have about 86 billion.
Which sounds like a small correction, but a difference of 14 billion neurons is equivalent to the entire brain of a baboon.
Wow.
It is not a rounding error, but the true paradigm shift from her soup method wasn't just the total number. It was discovering exactly where those neurons are hiding.
Wait, if the wrinkled cerebral cortex is the part we associate with deep thinking and philosophy and language, why on earth does the tiny cerebellum at the back of the neck contain 80% of the brain's cells?
I know it sounds entirely backward.
Like, the cerebellum only makes up 10% of the brain's volume, but it holds nearly 69 billion of those 86 billion neurons.
It forces us to decouple physical size from biological processing power. The cerebellum, or little brain, is primarily responsible for motor control, balance, and timing.
Not philosophy.
No, not philosophy.
Mm-hmm.
But think about the sheer computational density required to coordinate the incredibly complex microsecond by microsecond timing of human movement in a gravitational field.
Oh, that makes sense.
Yeah, every time you reach for a cup of coffee, your cerebellum is running massive real-time physics calculations. The cortex might come up with the abstract plan to get the coffee, but the cerebellum is packing the raw, dense circuitry required to actually execute that movement smoothly.
Okay, so we have the outer cortex for the high-level processing and the cerebellum coordinating the movement. But beneath the cortex, deep in the center of the brain, is the older, primitive engine room.
The deep structures.
Yeah. If we peel away the cerebrum, we hit the brainstem. And in evolutionary terms, this is like the oldest hardware we have.
It is. The brainstem is the foundational stalk that connects the brain to the spinal cord. It controls the absolute autonomic basics of keeping you alive, breathing, your heart rate, blood pressure, and your baseline level of consciousness.
And sitting right on top of that life support system is the diencephalon, which houses the thalamus and the hypothalamus. Now, I always like to think of the thalamus as the ultimate internet router for your nervous system.
That's a really good analogy.
Right, because every piece of sensory data coming in from your eyes, your ears, your skin, everything except your sense of smell, it all hits the thalamus first. The thalamus filters all that raw data, decides what is actually important, and routes it up to the correct processing departments in the cortex.
Exactly. And without that router filtering the signals, your cortex would just be instantly overwhelmed by sensory static. You wouldn't be able to function.
And right beneath that router is the hypothalamus, which is barely the size of an almond, but it dictates your most fundamental drives.
It's small but mighty. It regulates hunger, body temperature, sleep cycles, and it interfaces directly with the pituitary gland to control your entire endocrine and hormonal system.
And then wrapping around these deep structures is the limbic system, which is kind of the seat of human emotion and memory generation.
Right. You have the amygdala, which acts as the brain's threat detection alarm, processing fear and emotional responses.
And next to it is the hippocampus, which, fun fact, doesn't actually store your memories, but acts as the factory floor required to form new, long-term conscious memories.
Yes, a crucial distinction. And finally, you have the basal ganglia, which are these clusters of neurons deep in the brain that help initiate movement and lock in our automated habits.
Now, the functional stakes between these inner core structures and the outer cortex are wildly different, aren't they?
Oh, absolutely. A patient can suffer a traumatic injury or a stroke and lose fist-sized chunks of their outer cerebral cortex.
And they survive.
They do. I mean, they will suffer localized deficits, maybe losing the ability to read or move a certain limb, but they survive, and their core identity often remains intact. Contrast that with the brainstem.
Stakes are higher.
Much higher. A lesion the size of a pea in the brainstem is often instantly fatal because it unplugs the autonomic machinery keeping the heart and lungs running.
Wow. Now, we understand this layout today, but historically, the functions of these deep and outer regions were only mapped because of horrific accidents.
Tragic teachers, as they're sometimes called.
Yeah. And the most famous tragic teacher of neuroscience is Phineas Gage. So in 1848, Gage was a reliable, well-mannered railroad foreman.
Oh, the accident.
Right. While he was tamping down explosive powder with a heavy iron rod, it detonated.
Yeah.
The 3-foot rod shot up through his left cheek and blew completely through his left frontal lobe.
And Gage miraculously survived the physical trauma. He was actually walking and talking shortly after the accident.
Which is mind-blowing.
It is. But his physician, John Martin Harlow, documented a profound psychological transformation. Gage became impulsive, profoundly profane, and incapable of executing plans.
Like his entire personality changed.
Exactly. He lost his social inhibitions entirely. And Gage provided the foundational evidence that the frontal lobe isn't just an arbitrary processing center. It is the physical, biological substrate of executive judgment and personality.
And then, almost a century later, in 1953, medicine learned another brutal lesson from a patient named Henry Molaison.
Right, known for decades in medical literature simply as HM.
He suffered from these debilitating seizures, and a surgeon named William Beecher Scoville attempted to cure the epilepsy by surgically removing the medial temporal lobes, including both hippocampi.
Which did improve the seizures, but the surgery induced profound anterograde amnesia.
Meaning he couldn't form new memories.
Exactly. HM lost the ability to form any new conscious memories for the rest of his life. He was basically frozen in time. He would meet the same researchers every single day for decades and greet them as total strangers.
Okay, here's where it gets really interesting. One of those researchers, Brenda Milner, gave him a task, tracing a complex shape while only looking at his hand through a mirror.
Right, mirror tracing. It's a highly counterintuitive motor skill.
Super hard. And HM would attempt it, and the very next day, he would have zero recollection of the room, the researcher, or the mirror. But when he picked up the pencil, his hands were better at tracing the shape.
Which shouldn't be possible, right?
Exactly. If his memory was completely erased every day, how did his hands get better at it?
That specific contradiction fundamentally rewrote our understanding of the brain. HM proved that memory is not a single, unified folder in the brain.
It's not just one hard drive.
Exactly. It consists of anatomically distinct parallel systems. Because his hippocampus was removed, his declarative memory, the ability to consciously recall facts, names, and events, was gone forever.
But
His basal ganglia and his cerebellum were completely intact. Those structures govern procedural memory. So his brain could still unconsciously learn and automate a physical motor habit, even though his conscious mind had no record of it.
it's an incredibly elegant system. But treating the brain as a map where every function lives in one isolated coordinate has also led medicine down some incredibly dark paths.
Yes. If we connect this to the bigger picture, the entire history of neuroscience is basically a tension between localization, Broca's idea that specific regions have specific jobs, and holism, which is the idea that complex functions require the entire brain working as an integrated network.
Right. And in the early 20th century, a researcher named Carl Lashley spent decades trying to pinpoint the exact location of a memory.
He trained rats to run mazes, then surgically removed different portions of their cerebral cortex to find exactly where the maze memory lived.
But he never found a specific memory address, right?
No, he didn't. Lashley discovered that the rat's memory loss was determined by the amount of tissue he removed, not the specific location.
He called this principle mass action.
Exactly, mass action, suggesting memories are distributed across broad, overlapping networks rather than just stored in a single spot.
So Lashley was pointing toward this crucial truth about interconnected networks. But mainstream medicine largely ignored the nuance of networks, clinging instead to extreme localization.
They assumed that if the frontal lobe controlled behavior, then behavioral problems could be fixed simply by disconnecting it.
Which brings us to the prefrontal lobotomy.
A very dark chapter.
Yeah. In 1949, Portuguese neurologist Egas Momis actually won a Nobel Prize for developing a surgical procedure that severed the connections to the frontal lobes to treat severe mental illness.
And then an American doctor named Walter Freeman took it even further.
He completely stripped away the operating room entirely and popularized the transorbital lobotomy. Freeman would insert an ice pick-like tool through the patient's eye socket and just blindly sweep it back and forth to scramble the frontal lobe connections.
It took minutes, often without anesthesia.
And roughly 40,000 of these procedures were performed in the U.S. alone by the 1950s, leaving thousands, including Rosemary Kennedy, severely disabled. I mean, how did a genuine anatomical insight that the frontal lobe governs judgment become a license to basically scramble people's brains with an ice pick?
Because they confused a regional map with a complete understanding of a complex system. They viewed the brain as a static machine.
Like a car engine.
Right. If a machine is glitching, you isolate the broken part and snip the wire. They completely fail to understand that severing those vast white matter networks doesn't just fix a symptom, it fundamentally dismantles the entire human being.
But we now know the brain is anything but a static machine. It is an incredibly dynamic organ that physically alters itself based on how you use it.
Absolutely.
Which means we should probably bust a few more myths while we're at it. First, the idea that we only use 10% of our brain.
Oh, yeah, completely false. Brain imaging has proven that practically the entire organ is active over the course of a normal day.
And second, the left brain equals logical, right brain equals creative personality myth.
Also false. While certain functions like language do lean heavily to the left hemisphere, there is no evidence that people rely predominantly on one side, or that it dictates your personality type.
But what is definitively real is neuroplasticity.
Yes, the physical architecture of the brain literally reorganizes itself in response to learning and experience.
And I love this. The most striking proof of this came in the year 2000, looking at London taxi drivers.
The famous cabbie study.
Yeah. So to become a licensed cab driver in London, you have to pass this brutal exam called the Knowledge, which requires memorizing the layout of roughly 25,000 city streets.
So neuroscientist Eleanor Maguire put these drivers into MRI machines and looked at their hippocampi, the region involved in spatial memory.
And she found that the drivers had significantly larger posterior hippocampi compared to the general population.
And the longer a driver had been navigating the streets of London, the larger that physical structure grew. Intensive, prolonged use of spatial memory literally recruited and built up physical gray matter.
That's amazing. Learning literally physically stretches the brain like a muscle.
It does. But that discovery fuels a massive, ongoing fight in the scientific community today. We know the existing neurons can form new connections, but does the adult brain actually generate entirely new neurons?
Right, neurogenesis.
This raises an important question about how science actually operates at the frontier. The debate over adult neurogenesis is entirely unresolved.
Wait, really? We still don't know.
We really don't. In 2018, the prestigious journal Nature published a rigorous study showing no evidence of new neuron production in adult human hippocampi.
Okay.
But that very same year, the journal Cell Stem Cell published an equally rigorous study showing evidence of ongoing neurogenesis throughout human life.
So they totally contradicted each other.
Exactly. They used different methods for preserving the tissue and reached entirely opposing conclusions.
So we are literally still trying to figure out if the hardware is fixed or constantly replicating. So what does this all mean? Why does this detailed map of lobes, brainstems, and plasticity matter to you listening to this right now?
Well, because mapping these specific regions dictates exactly how we treat disease and save lives today.
Like with strokes.
Yes. When a patient suffers a stroke, emergency responders use the FAST protocol looking for face drooping, arm weakness, or speech difficulty.
And that protocol works because we know specific blocked arteries will starve very specific, localized, functional areas of oxygen, right?
Precisely. The symptoms instantly tell the doctor which part of the map is dying, allowing them to deploy clot-dissolving drugs in a tight time window.
And this map is also how we track neurodegenerative diseases. Alzheimer's disease specifically attacks the hippocampus and temporal lobes early on, which is why the inability to form new memories is that hallmark early symptom.
And Parkinson's disease disrupts the basal ganglia, but it specifically originates from the death of cells in a tiny, deep brain structure called the substantia nigra.
Which produces dopamine.
Right. The substantia nigra acts as a crucial factory producing dopamine. When that factory dies, the basal ganglia loses its fuel, resulting in the rigid tremors associated with Parkinson's.
And understanding those specific mechanisms allows us to actually intervene. We are now developing brain-computer interfaces based on this map.
It's incredible technology.
By mapping the motor cortex, surgeons can implant electrode grids into paralyzed patients, allowing them to move computer cursors or robotic limbs simply by firing those specific neurons.
And for Parkinson's, we use deep brain stimulation, surgically implanting pacemakers directly into the basal ganglia to supply the missing electrical rhythm and instantly calm the tremors.
It is wild to think about the journey we've been on. From an Egyptian surgeon noticing paralyzed limbs in 1600 BCE to implanting electrodes deep into the basal ganglia today, humanity has spent thousands of years charting this biological territory.
It really is a grand tour.
We've moved from believing our heads were just radiators for our hearts to discovering we have 86 billion neurons, mostly packed into that tiny cerebellum, to realizing the brain is a constantly shifting network of specialized regions.
It is the ultimate puzzle. We have spent centuries using our brains to systematically dissect, label, and map our own brains, unlocking the mechanics of memory, movement, and consciousness along the way.
It's almost a paradox. And here's a final thought to leave you with. Because of neuroplasticity, because your brain physically alters its synaptic connections to encode new information, the simple act of listening to this deep dive has physically changed you.
That's very true.
The 1.3 kilogram mass inside your skull is structurally, biologically different right now than it was 20 minutes ago. It is constantly reshaping itself to reflect the world you interact with. Keep that in mind as you go about your day.